In this discussion, we'll talk about differential expression and how we assay the levels at which different genes are expressed in different samples. A little bit of a review. Well, we've talked about some of these concepts in previous lectures. So first off, the thing that I wanted to review is sequencing depth. So basically, when we say sequencing depth, we mean how many random pieces of DNA am I going to sequence? And remember that when we talked about genomes, we talked that we wanted a sequencing depth where our total coverage, so the total number of base pairs that we read, was about 50 times the estimated size of the genome. And the reason that we need 50 times is because it is a random sample. And so the very first random sample you take is guaranteed to be unique. It's new, never been sequenced before. And then the second one is almost certainly new. It's very unlikely that in a genome of like 3.2 billion base pairs, like the human genome, that your first two sequences by chance would overlap. And so we continue to do that, and we do hundreds of billions of reads for a genome. And so for our, well, maybe, a little bit more than 100 billion. So for instance, if we had a genome of 3.2 billion base pairs like the human genome, we'd want to do about 150 billion base pair reads altogether. So if our reads were 100 base pairs in length, we would do, however many 100 base pair reads until we got to a total of about 150 base pairs total. And again, the reason for that is because of the random sampling. As we add reads to our sample size, more and more reads, then they're going to be somewhat redundant, which to some extent is what we want, right? We want them to be a little bit redundant so that we can find their overlaps, line them up, and then assemble the entire genome. But the more reads we get, the more we are just resampling things that we've already got before. And so that's where sequencing depth comes in. And so at 50x, we might have some areas of the genome that just because of chance are covered more than 100 times. We might have some that are only covered a couple of times. And inevitably, just again, it's that random sampling thing, we're going to get some small pieces of the genome that maybe never get sampled at all. And to some extent, the law of diminishing returns applies here, because as we put more and more resources, our samples are larger and larger and larger. The more reads we do, the less chance there is of sampling something new, because we've already covered 99.999% of the genome. So the chance of those final reads being something new are fairly low. And so we have fairly good coverage for most of the genome, but maybe poor coverage and a few little gaps even in some areas. Now, so the rule of thumb for genomes is 50x. But calculating the coverage depth, how much sequencing we need to do for transcriptomes, is a little less straightforward. So here are some of the things we need to consider. Number one, the total number of base pairs represented in a transcriptome is smaller than the genome. And for eukaryotic organisms, it's much, much, much smaller. So for instance, in the human genome, only about 1.5% is protein coding. So in a transcriptome, we're looking at even if every single gene was expressed in a sample, which not every single one is going to be in most of our samples, but even if every single gene was expressed, we would only have about 1.5% of the total genome size. So initially you'd think, oh, instead of 50x, I only need to do 1.5% of that coverage to have the same level of coverage, which would be true. However, there's an additional problem, which is that every gene is expressed at different levels. And so typically what happens is we end up with a gene that might be expressed at really, really, really high levels. For instance, if we have a sample of pancreatic cells that are expressing insulin, maybe even 90% of all of our transcripts are the insulin gene, because it's just making tons and tons of transcripts and pumping out insulin. And then the other 10% of all the samples would be all the rest of the genes put together. That's maybe a little bit of an over-exaggeration, but we still could have a very, very large percentage representing just one gene or a handful of genes, depending on our tissue type. And then other genes may be there and being used, but at such low levels that we would need to have a really, really good coverage, of fairly deep sequencing, to be able to capture those very lowly expressed transcripts. So there are some rules of thumb just about general amounts of reads that need to be done, but the main take-home message that I want you to know is there is no fixed rule because transcriptomes are more dynamic and variable than genomes. Okay. Now, one other thing that is an important logistical and experimental design consideration when we're doing transcriptome sequencing. And to some extent, this can also apply to genomes, but because genomes are larger, it's not quite the same, particularly for eukaryotic organisms. So if you need to, you can go back and review the Illumina processing for next-gen sequencing. But as part of that process, we take our original sample, which is labeled here as an insert. That's the small piece that we've cut out of our transcriptome of our genome. And we put adapters on the end. And these adapters have amplification elements that allow them to bind to the silicon plate and then be amplified and sequenced. But in addition, they also have these things that we call indexes, or indices is another plural, but an index is put onto each of those samples. And so what we can actually do is that we can put different indexes onto our different samples. So if I've got sample number 1, which is maybe a treatment that's, I've added chemicals to something, and then sample number 2, which is the control. I could do those as I'm creating the libraries for sequencing in separate tubes and add a different index on the first sample that is unique and distinct from the index on the second sample. And then the reason that we do this is so then we can pool them together. We can combine them into a single sample that is all put together onto the next generation machine to do the sequencing. And the reason that we do that is often the volume, the number of reads that we can get off of any single lane on our machines is way, way more than we may need for a single transcriptome. And so I might be able to do 10, 20, even 30 different transcriptomes on a single read, on a single sample on my next generation sequencing machine. And so if each one has a unique adapter, then I can just read them all. And then afterwards, the computer can sort them out based on that index. And so just be aware of how that index works. And we do it so that we can maximize the number of samples, still have adequate coverage for each individual sample, and get the most bang for our buck, so to speak, to be efficient about how we are using our resources as we're doing this sequencing. Okay? Now, once that sequencing is done, the assembly is very similar to the way that we assemble a genome. But again, remember, instead of large chromosomes, scaffolds, and then chromosomes, we're going to get many thousands, maybe even tens of thousands of particular transcripts. Okay? Now, the other thing that we need to remember when we are sequencing transcriptomes for differential expression is we need statistical power to say that, yes, I'm certain that this is an accurate measure of the level of expression. And to do that, we need more than one sample. So for instance, if you were going out into a field and you wanted to determine the average size of a individual in a population, let's say you want to know what the average adult size of white-tailed deer are. If you went out and picked one individual, that may or may not be a good average, a good representative of the adults in that species, right? Especially if there's lots of variability based on genetics or based on environment. You might have to sample 100 or 200 individuals randomly across a wide distribution to get a good feel so that, okay, I really have a good confidence in what the average size is. And depending on your sample size and the strategy for sampling, you would then have some margin of error, some variance, right? And so you could represent both an average size and then variance or margin of error of how confident you were that that was really an accurate represent of the average size. So the same thing is true for expression of genes. If we just take one sample and go through the process and measure the level of expression for a gene, we might have some error that's been introduced by our methods. There might be some random variation in that sample. So we want to be pretty confident in it. So to do that, we need multiple samples, or what we call replicates. So for each of our treatments, sometimes we'll use the term sample, but for each of our treatments, we want to do multiple replicates. And each of these would be sampled independently. RNA would be extracted independently. Each of the libraries would be created independently, and then they would each be sequenced independently. So each of them represents the exact same treatment, but a extra sample or an extra replicate. Okay, so here's an example, a very simple experiment. Let's say we wanted to know differential gene expression from a normal set of skin cells versus a cancerous set of skin cells. And so I would take 3 independent samples of healthy skin. I would extract and sequence their RNA and assemble a genome, and I would call it replicate N1, N1 for normal cells. And then I'd have an N2 and an N3. Each of these, in theory, should be very, very similar or identical. Right? These should be identical treatments. The skin should come from the same general location at the same time of day. We want these to be as similar to one another as possible, but to give us multiple independent replicates. And then I do the same thing for cancer cells. Again, as identical as possible, but 3 independent samples, all sequenced and assembled independently. Then, to do our analysis, we take all of these replicates. And because these replicates come from the same genome, right? So cancer genome is essentially identical to a, there are some small mutational events, but they're largely identical to normal skin cells. And so I can take all of these replicates and I can assemble them together in one large global assembly. And so for each transcript, I might find many, many reads that all go together to create this one transcript. Some of these reads might come from replicate number C1, some of them from C2, and we're hoping that in that treatment, they're largely going to be the same or at least very similar. But some of them might come from these other ones. So that global assembly is all of our replicates together. And so I end up with this big transcriptome that's been put together from all of my replicates. So again, the replicates, typically 3 is very, very standard. Some people will do more. You've got a little bit more power and your margin of error, right? That variance in your samples might be a little bit smaller. We might be able to have a little bit more power to determine, yet, yes, this is actually a very, very good measurement if we do more. But again, there's a kind of a law of limited returns. If you do, same thing if you were going to do a survey of the deer, right? If you sampled 100 deer randomly, then you could maybe do 10 times as much work and only get a little bit more confidence if you did 1,000 deer versus 100 deer. So there's kind of a balance in how many resources I want to put into and what level of surety I want about my results. So typically 3 replicates is very standard and is usually considered sufficient to capture most of, to get an accurate and powerful measurement of the average expression for each gene. So we end up with a global assembly with tens of thousands of genes that have all been assembled from reads from these different replicates. So then what we have to do is we have to match each of these replicates to the different reads that are in our global assembly. So for instance, the very first one that we have has been assembled from all these different reads, and I would go in and I would count how many of these reads come from replicate normal cell number 1. And I count them all up and I say, oh, 230 of these reads come from it. And I do the same thing, and hopefully we would get something consistent like this, where there's going to be a little bit of variation. Some of this might be, error in our methodologies. There is some random sampling error in it also. Might be a little bit of environmental variance if our treatments were not identical, or just, there is some stochasticity, some randomness, even into identical cell samples. But if it is something measurable, and going on the same thing in all of our samples, they should be about the same. Then I look at my cancer treatments and notice that in this example, this is hypothetical, but in this example, I have maybe about, I don't know, that would be 4 times 5, maybe about 20 times the level of expression in our cancer cells for this first transcript, where 4,000, nearly 5,000 of these reads came from replicate C1, another 5,000 came. So in total, this one would have, about 15,000 plus maybe another, close to 1,000, maybe a little less. So maybe about 16,000 reads went into making up this transcriptome. But notice many more of those reads came from the cancer samples than came from the normal cell samples. So I could go in and say, with some statistical margin of error, say that there is about 20 times the level of expression for this gene, this transcript, in cancer cells versus normal cells. And then I would just repeat this for all of my tens of thousands of transcripts that were present in my global assembly. And I would get out for each transcript, is it expressed at the same level in treatment number 1 versus treatment number 2? And if there's a huge margin of error, so if I'm getting normal cells at 4,000 and then normal cell sample 2 at 200, then that's, I don't have good power. I can't really say that there's something different because I'm just getting too much variation from my different replicates. But if everything's done carefully and well, then we're going to have very consistent results across the different replicates within the same treatment. And so the end result is a huge amount of data. For all of my tens of thousands of transcripts, I have data about expression levels in one treatment versus the other, also data about the levels of variation within the different replicates in those treatments. So to try and summarize this massive amount of data, there are a couple of different things that we do. We could look first for target genes. So if we know that there are particular genes that have been indicated as important in the process of cancer or cellular division, cell cycle, these are the things that are impacted by cancer, I could say, oh, look, here are 100 genes that I'm really interested in. We could go in and do those target genes. But maybe there's some genes that are unexpected that we maybe would not know ahead of time to look for, but might still show some different levels of expression. And so we could look especially for the outliers. And one way to do this, we can visualize it, and then we can also identify individual transcripts with two different analyses. And I've talked about these briefly in the last discussion, but we'll just go over them a little bit more. A volcano plot is a plot that along one axis, the x-axis, shows kind of a normal level of expression here in the middle, and then a upregulation of expression in sample 1 and a downregulation of expression in sample 1. So these are the genes that say in the cancer cells would be upregulated. These are the genes in the cancer cells that would be downregulated. And then along the y-axis here is the total level of expression. So these genes that are really, really high have either high downregulation or high upregulation. And so we've got a group of genes, a cutoff here for significance, but a smaller group of genes that are very highly upregulated or downregulated. And so we could take maybe the most, the 100 or so most upregulated genes in cancer cells and begin to study them, or the 100 or so downregulated genes and begin to study them and see what their role is, how important are they in this process of disease development in cancer cells. And so if we see a volcano plot that's very low and not very spread out, that means that our two different samples are very, very similar with only small levels of different expression. But if it's very, very high and very spread out wide, that means that we have a big difference in expression levels of different genes. Now, the other graphical representation that is commonly used is called a heat map. And heat maps by color show upregulation and downregulation, but instead of just comparing two different samples, we can compare all of our samples. So if we have 9 different samples, we could compare all of them and find the genes that were upregulated and downregulated. We could even find groups of genes. Maybe these are all similar in the amount of expression level, or they're all closely related to one another. They're all part of the same gene family. And notice we kind of have this block of blue that in sample 1, 2, and 3 are all blue, so they would be downregulated. And here we have a block that generally, a block of different genes that are generally all upregulated. And so this tells us whether there are distinct patterns in our sample or whether it's just kind of a random gene here and there that is upregulated and downregulated. So both of these are useful things. Not only do they give us kind of a big picture overview of patterns of expression, but then they also allow us to identify individual genes that might be of particular interest. Now, the last thing, again, a little bit of a review, but I just wanted to go over it one last time, make sure it's clear in your head, is that this is only one methodology to assess gene expression. The other main methodology, which is much less of a global approach, right? We can't do every single gene. Well, we can, but it just takes tons and tons and tons of resources. But what we can do is we can verify, let's say these 100 genes that are downregulated and the 100 genes that are upregulated. Maybe we'd like to independently verify that, yes, that's an accurate measurement. They really are either up or downregulated. So what we could do is we could validate that expression using an independent methodology. And the most common methodology that is used is qPCR. Now, I'm not going to go into details about qPCR. If you are not familiar with it, you should probably Google it or look up a little short video about what qPCR does. But basically, it is a PCR method where we can estimate the amount of starting transcripts based on when they begin to amplify. They'll all end up saturating at the same level, but depending on when this saturation curve begins the logarithmic growth, we can estimate an initial sample of the size. So we could take an RNA sample and amplify a gene from that. And then based on when this begins to amplify, we know we can estimate the total number of transcripts in that beginning sample. And so this can independently verify these these numbers that we're getting out. Like we say one is 10 times or 20 times more common than the other one. Well, we could verify that, yes, indeed, our qPCR results seem to corroborate, substantiate the levels of expression that are there in those two different samples.